Okay, so genetic technology and medicine. Genetic technology allows products specific to humans to be made. We've already looked at the advantages of producing human insulin by recombinant DNA techniques. Other human proteins are produced by similar techniques. For example, human growth hormone, thyroid stimulating hormone, factor 8, a blood clotting protein. So, kind of similar techniques are used. There are advantages of using bacteria and cultures of mammalian cells to produce these proteins. These cells have simple nutritional requirements, large volumes of products are produced. The production facilities do not require much space, and the process can be carried out almost anywhere in the world. There are few practical and ethical problems because proteins do not have to be extracted from animal sources or by collecting blood from many donors. The disadvantage of using bacteria to produce human proteins is that bacteria do not modify their proteins in the same way that eukaryotes do. It is much better, therefore, to use eukaryotic cells to produce human proteins. What it said there, suppose we use that process to produce insulin to produce another type of protein, but the bacteria can only produce the protein. But once the protein is formed, the protein may need to be modified. But bacterial cells cannot do that. That's because they are not complex. The genes are small in number compared to eukaryotics. For the complexity of the genes of eukaryotes, they can carry much more function. So this is a disadvantage of using bacteria, but it can be solved by using eukaryotic cells. Genetically modified hamster cells are used by several companies to produce factor 8. This protein is essential for blood clotting, and people who cannot make it are said to have hemophilia. The human gene for making factor 8 has been inserted into hamster kidney cells and ovary cells, which are then cultured in fermenters. In fermenters, we use the nutrients and a suitable environment where the kidney cells or ovary cells will, secrete the product which you need. In this case, it is factor 8, and it can go on forever, almost like what we have studied in insulin production by using bacteria. The cell constantly produces factor 8, which is extracted and purified before being used to treat people with hemophilia. These people need regular injections of factor 8, which before the availability of recombinant factor 8 came from donated blood. Using donated blood carried risks of infection, for example from HIV. Recombinant factor 8 avoids such problem. High yields of the enzyme adenosine deaminase, which is used to treat severe combined immunodeficiency disease, are made by genetically modified insect larvae, the cabbage popper moth caterpillar. Well, the gene for producing this enzyme is inserted into the larva, and then the larva produces the enzyme, which is in a fermenter so that it can go on forever, which is then purified and collected. Almost the same thing, it's the same thing but in different scenarios. This enzyme is administered to patients while they're waiting for gene therapy or when gene therapy is not possible. Some proteins are even produced by transgenic animals. Sheep and goats have been genetically modified to produce human proteins in their milk. Human antithrombin is produced by goats. This protein is used to stop blood clotting. Human alpha antitrypsin is produced by sheep. This is used to treat people with emphysema. So yes, we can you genetically modify a animal, like maybe from the birth, like when they were just an embryo, or maybe after they have born. We can change the cells that we can insert the gene for producing these products into the cells of the milk producing gland, and the milk will also be produced along with the product which you need. So yeah, there are a lot of processes by which genetic engineering can be used to produce medicine. Okay, so genetic screening. Genetic screening is the analysis of a person's DNA to check for the presence of a particular allele. Well, basically the allele which causes diseases. This can be done in adults, in a fetus, or embryo in the uterus, or in a newly formed embryo produced by the in vitro fertilization. Well, we know that fetus is like the developing baby, and embryo is like only quite many groups of cells that forms, which will become the fetus. An adult woman with a family history of breast cancer may choose to be screened for the faulty allele of the gene BRCA1 and BRCA2, which considerably increase an individual's chance of developing breast cancer. Should the result be positive, the woman may elect to have her breasts removed before such cancer appears. So genetic screening helps us to identify the genetic disease that might occur before it shows up or the symptoms shows up, so we can take preventive measures. And we know how to genetic screen. We know electrophoresis and microarrays. We've learned this, so really useful thing. In 1998, the first designer baby was created, officially known as pre-implantation genetic diagnosis. The technique involved mixing the father's sperm with the mother's egg in the dish that is normal in vitro fertilization produce. Like, the reproduction is done artificially, not the naturally how it happens. It was the next step that was new. At the 8th cell stage, one of the cells from the tiny embryo was removed. The DNA in the cell was analyzed and used to predict whether or not the embryo would have a genetic disease for which both the parents were carriers. It could be that the parents had a genetic disease which could have been passed to the child. So if the child was born, then he would be born with the disease. But if we could screen the embryo first, we can know that this embryo will form into a child with the disease, and so we may discard the embryo. Yeah, this will reduce the number of genetically diseased child, which is the genetically diseased child, which the disease occurs because of from birth, which are inherited from their parents. Okay, the DNA in the cells are analyzed to predict whether or not the embryo would have genetic disease for which both the parents are carriers. An embryo that was not carrying the allele that would cause the disease was chosen for implantation, and the embryos that did have these alleles were discarded. Implantation means putting the embryo into the uterus of the mother so that the embryo can grow, because embryo needs to grow into the fetus. It don't happen in a petri dish or anyhow. It needs to happen inside the mother, inside the womb. Yeah. Since then, many babies have been born using this technique. It has been used to avoid pregnancies in which the baby would have had Duchenne muscular dystrophy, thalassemia, hemophilia, Huntington's disease, and others. All these are basically genetically diseases which are inherited from parents. In 2004, the first used in the UK to produce a baby was a tissue match with an elder sibling, with a view to using cells from the umbilical cord as a transplant into the sick child. So basically the child was sick, so the tissue from the child was taken and it was used to make an embryo which was implanted into the mother, so that when the fetus formed, cells from the umbilical cord of the fetus can be used to be transplanted into the sick child. Because if we take the cells from other donors, then the immune system would identify them as a foreign cell, and so they would have destroyed the cell. But if it was produced with the same genetic material of your cell, then it is not a foreign cell. It is they will identify it your cell. So this was a way to make your immune system full and think that those cells were actually yours. Well, they were actually yours in a way. For some time, genetic testing of embryos has been leaving prospective parents with very difficult choices to make. If the embryo is found to have a genetic condition such as Down syndrome or cystic fibrosis, the decision about whether or not to have a termination is very difficult to make. Now the advances in medical technology have provided us with even more ethical issues to consider. Well, we will consider because even though it is an embryo, it is their child. They may be emotionally excited that they would not want that baby or that embryo to be discarded. So we will look at the ethical issues about genetic screening later. Okay, so the ethics of genetic screening. In 2004, UK allowed an embryo to be chosen that did not have an allele for a genetic disease and also one that did have a tissue type that would allow a successful transplant into a sick elder brother or sister. Okay, good. These are all medical reasons. But it did not allow the addition of an allele to an egg, sperm or zygote. Well, let's imagine that we have identified an allele that would cause us to be very much intelligent, just like Einstein. If we could have identified that allele, and if there was an allele that caused this reason, we could have inserted it into eggs or sperms or even a zygote. Zygote, when a sperm and an egg combines, it forms a zygote. And then the baby would have been as talented as an Einstein. I'm just kidding, it may not happen, but addition of an allele was not possible. The baby had to be natural. A line had to be drawn somewhere, but feelings can run high. Many people believe that the law is allowing too much, while others think that it should allow more. Other countries have different attitudes and different regulations. There is still controversy over other long-established outcomes of genetic screening. For example, a fetus can now be screened for a genetic disease while in the uterus using amniocentesis or chronic villus sampling. Now let's see what are these. The parents may then decide to have pregnancy terminated if the embryo is found to have genetic disease. Like when the fetus is in the belly or womb of the mother, it may be screened for genetic diseases, and if they were positive, the parents can terminate the pregnancy. Well, however, there have been cases where decisions have been made, even though the defect has been a relatively minor one, with which the child could be expected to lead a fairly normal life. Some parents have decided to terminate pregnancies simply because the child is not the sex they want. The gender they want. Maybe they wanted males, but female was born, so they terminated it. This is ethically wrong. This is an unethical thing. They have also used PGD to select the sex of the embryo they want to choose to implant, which is really biased and it is wrong, unethical. Many think that this sex pre-selection, as it is called, is totally unethical. Okay, now let's see what's amniocentesis. Amniocentesis is used to obtain the sample of amniotic fluid at 15 to 16 weeks of pregnancy. What's an amniotic fluid? See this baby? It is in a sac inside the fetus, and the sac is called the amnion. Now, inside the amnion, there is this amniotic fluid in which the baby lies. Okay, so a sample of the fluid is taken and screened for genetic diseases. Okay, this is called amniocentesis. Various tests can be carried out on the sample to check the health of the fetus. Most amniocentesis samples, however, are to look for chromosomal mutations. Ultrasound scanning can be used to visualize the fetus and locate the position of the placenta, fetus, and umbilical cord. The umbilical cord is this cord which is connected to the nerve of the baby, and this is the placenta. This part is called the placenta. Okay, it is a placenta. Placental walls from which nutrients or waste materials are exchanged between the mother's body and the fetus. So we use ultrasound to identify the position of the fetus, umbilical cord, and the placenta. Why we do that? A suitable position point for the insertion of the hypodermic syringe needle is chosen, and this is marked on the abdominal skin surface. Well, hypodermic needle means very thin needle, and abdominal skin surface means the belly of the mom, the skin of the belly. Generally, this position is away from the fetus, umbilical cord, and placenta. These are the amnion. The fetus is in the belly, and that's only the skin of the mom. Chorionic villus sampling has been carried out between 10 and 13 weeks of pregnancy, so it allows parents to get an earlier warning of any genetic abnormalities in the cell than it is possible with amniocentesis. It is more easier to identify these instead of amniocentesis. A small sample, a part of the placenta called the choroid, is removed by the needle. There is a part in the placenta called the choroid is removed by the needle. The needle is narrow. Because else it may damage the baby, or the amniotic sac may be damaged, and that's all the amnion fluid burst out, leaks out, and the baby is gonna die because it don't have any amniotic fluid. The procedure is monitored by ultrasound scanning. We have to scan to see if the needle is in the right position that we need, or has it affected the fetus or the baby. So we need an image. We cannot cut the belly and see this, so we use ultraviolet ultrasound. This is how we carry out the procedure. Amniocentesis, chorionic villus sampling has a small increased risk of miscarriage. It has been estimated that miscarriage rate is increased by 1 to 2%. This is slightly greater risk than for amniocentesis, but before 15 weeks, chorionic villus sampling is probably less risky than amniocentesis. This is just a few good information which you may need. Thalassemia is a blood disease similar to sickle cell anemia. It used to be a common genetic disease in countries around Mediterranean, Cyprus, Greece, and southern Italy. The incidence of the disease has decreased significantly over recent years as a result of genetic screening and giving advice on couples who are identified as carriers of mutant alleles. Because of genetic screening, we can terminate the pregnancy of the diseased child, and so the child will be born into the world, and thus the number of people with the disease is decreased. Only healthy babies are born. When the fetus was identified as having inherited that disorder, couples received advice about the possibility of terminating the pregnancy. Terminating pregnancies for medical reason rather than any other is known as therapeutic abortion. By any other, I mean if you wanted a boy or if you wanted a girl, this. If for these reasons it was terminated, a healthy embryo or healthy fetus was terminated, then that is not therapeutic abortion. Only if for medical reasons, maybe it is the carrier of a disease, genetic disease, then the pregnancy may be terminated, and that termination is called therapeutic abortion. Huntington's disease is a late onset disease. Symptoms do not usually appear until middle age, by which time people have usually already had children. There is no cure for this disease, and the treatments available can only alleviate the symptoms. People in families with Huntington's face a dilemma. Should they have genetic tests to find out whether or not they have the dominant allele for the disease? They also pose ethical dilemmas. Would you rather be told that you are at high risk of developing the disease, even though nothing can be done about it, or live in the uncertainty of not knowing? Is it a good idea to have the information before you start a family? Decisions are made even more difficult by the possibility that the person with the dominant allele for the Huntington's disease may live their whole life completely free of the disorder, as it sometimes does not develop. So like maybe those parents did not perform genetic screening, and so the baby had Huntington's disease. And since it appears in the middle age, like in that time, those parents may have also like taken babies who may inherit the disease. And so like a lot of ethical questions, ethical dilemmas stand for that purpose.